Hey, another murder occurred. We need you to process this DNA as quick as possible. We think it's the killer.
Okay then.
The aluminum foil mylar bag contains 6 tubes, the 2X restriction digest master mix, 3 suspect DNA samples, sample A, sample B, and sample C, 1 unknown DNA sample collected from the crime scene, a DNA ladder for the gel electrophoresis. When not in use, keep all of these tubes on ice. Additional required materials are ice, heat block or water bath and centigrade thermometer, microcentrifuge tubes and racks, vortex mixer, P10 and P100 micropipettes and tips, gel electrophoresis equipment, gel chamber and power source, gel electrophoresis consumables, 1% agarose gel, TBE, DNA loading buffer, running buffer, DNA gel dye, transilluminator, UV light box, or gel documentation system. A camera is optional. Use a permanent marker when labeling things. Before you begin your experiment, you should prepare your heat block or water bath. Turn the heat block or water bath on. Be sure that the temperature is stable at 37 degrees centigrade before use. Place the 2X restriction digest master mix on ice to thaw. Once it is thawed, it is vital that before opening the tube, to spin it in a microcentrifuge for 10 seconds at 6,000 RPM. Vortex it for 10 seconds, and then spin it again for 10 seconds at 6,000 RPM. Begin the experiment by labeling the microcentrifuge tubes, A, B, C, and U, for sample A, sample B, sample C, and unknown DNA sample. The DNA samples are enough for 6 groups, namely 6 of A, B, C, and U. Next, add 10 microliters of the appropriate DNA sample into each tube, using a new micropipette tip for each sample. Aliquot 10 microliters of the 2X restriction digest master mix into 6 tubes. Add 10 microliters of the 2X restriction digest master mix into each of the labeled tubes. Each tube should now have a total volume of 20 microliters. Mix the sample in by pipetting up and down or flicking with your finger. Arrange the tubes in a microcentrifuge, in a balanced arrangement in the rotor, with the hinges pointing up. Spin for about 5 seconds to force all of the liquid to the bottom of the tubes. Store the samples on ice until ready to use. Incubate the tubes at 37 degrees centigrade for 45 minutes in the preheated heat block or water bath. Remove the samples from the heat block and place on ice for 5 minutes to stop the digestion. At this point, if there is no time to continue, store samples at -20 degrees centigrade until the following lab period. To run the resulting samples through agarose gel electrophoresis, follow standard procedures using a 1% agarose gel. Use a DNA dye, such as Gel Red or Cyber Safe, which is added directly to the molten agarose. Fill the gel box with DNA running buffer so that it more than covers the gel. Add 2 to 5 microliters of loading buffer to each sample. Load 10 microliters of the latter into the gel, and at least 10 microliters of each sample. Make sure to note which lane contains which sample. Plug the gel box into the power source, red to red, black to black. Run the gel electrophoresis at approximately 120 volts for approximately 30 minutes, stopping before the loading dye runs off of the end of the gel. Once the gel has run, view it under a UV light exposure and record the results manually or by photography. Compare the bands. The DNA ladder can be used as a band size reference. Which subject sample, DNA, A, B, or C, matches the unknown sample collected at the crime scene? We hope that this video has provided you a clear idea of how to use the GenoSensor DNA Fingerprinting Kit 1. For more information, visit us at our website, www.genosensorcorp.com, or contact us at 480-598-5378. Thank you.